Then there's trial design. There are several different ways of constructing a trial. The paper that was assigned gives you the details, but I want to just talk about two of them in particular. One is the parallel design. That's the one we all know and love. You start off with a control group and a treatment group, and you follow them to the very end of the trial, and then you compare that. There's something else that's becoming more popular lately. It's quite controversial, and it's called the crossover design. And the idea behind the crossover design is that you use your pool of patients both as the control and as the treatment group. How do you do that? You basically split them into two, and one group you give the treatment and the other one you don't. You wait a while, and then you flip it around, and then you give the other group the treatment and you give the first group the placebo. And then you compare the two. Now, what's the attraction of this? Well, there are two attractions. One is that it looks like you're saving money. You're saving money because instead of having 2 N patients, maybe you can only deal with N patients and split them up and follow them throughout the period. The second attraction is that when it comes to accruing patients, when you're trying to attract patients, how are you going to feel if we do the first design, the parallel design, and you're in the placebo group? Now, you don't know that you're in a placebo group. You don't know what you're getting. But I can tell you that a recent colleague of ours who died of a particular kind of cancer, he was in a clinical trial. And I spoke to him a few weeks ago when he said to me, you know, things aren't really going well. I kind of know that I'm in the control group, and it really frustrates me. You know, I don't know if that was true. Maybe the drug was not effective. But you know that when you're engaging in a clinical trial, half of these individuals are not going to get the drug under the first design. And patients do not want to be part of that placebo group. These are individuals that are very sick, and they are looking at you for their last best hope. With the crossover design, you can say to all patients, you know what, everybody gets the drug. The only difference is when they get it. We're going to wait for some of you and give you the drug a little bit later to be able to tell whether or not you're going to get it. Now, it's controversial, so I'm going to come back to that. And but before that, question, Jacob?
I actually think the last point you made ties back to what you said earlier about having low enrollment of cancer patients in clinical trials. I think one of the big differences is, you know, if you're rolling in for a new vaccine or some sort of, you know, blood pressure regulation, you know, you can handle that. Whereas it's a worst case scenario for cancer patients. They've really exhausted all other options for approved treatments.
Right.
So, you know, why would somebody take a risk? You know, passing up, you know, they're actually just numbers-wise, there aren't that many available compared to someone who might say, okay, I'll volunteer for this vaccine. If it doesn't work out, there's not a huge personal risk.
Right, that's right. And I think that in those cases, there are examples of clinical trials where there aren't two arms. There's just the drug. Everybody gets the drug. And either they go by historical controls, in other words, what's happened in the past, or they'll look at surrogate endpoints where if you meet a certain target, that's enough to approve the drug. In certain cases, the FDA will actually take that into account. But you're absolutely right that for the standard 2-arm, randomized, balanced, controlled trial, individuals are not comfortable necessarily because they feel like, you know, they got a 50% chance of getting nothing, and they don't want to get nothing. But it's important that that control group is there, because if the FDA does not see real clinical efficacy, it will not approve a drug. And there are cases, maybe we'll hear about that a little bit later on, where it looks like there's a good drug out there, but because the clinical trial was designed poorly, the efficacy wasn't apparent from the data. And therefore, the FDA could not approve it. And once you get a negative decision from the FDA, you're dealing with damaged goods. It's not like you can just take it and try again a year later. All of the people who are involved are incredibly disappointed. Investors are really upset. And people leave, and they don't touch the compound. It's very difficult to resuscitate. It's not impossible. It's been done. But a negative decision is much, much worse than no decision. And those negative decisions are affected by clinical trial design. So let me give you a very cartoonish example using crossover. Crossover, this idea that you're allowing patients that aren't getting the drug to at some point cross over. The other reason that clinicians have started to engage in these practices is because it seems more ethical, right? Because everybody gets the drug, so you can tell them that. Everybody gets the drug, the only matter is when you get it. But there have been studies where the way that they make these crossover decisions is related to the severity of the disease. So in other words, they put you in a control group initially, and if you start to fade, if you start to suffer greatly, and you might benefit from this experimental drug, at some point, then you will be allowed to cross over, whereas if you're not doing so badly, you won't. What does that do? That basically is a selection mechanism that puts the sicker patients in the treatment group. And so if you put the sicker patients in the treatment group, odds are survival and efficacy are going to look lower than if you keep those two groups separate. And so because you want to do the right thing for a few patients, you might be destroying a drug for all other patients from that point forward. And so the ethics of crossover trials is quite complicated. It's not nearly as black and white as it might seem on the surface. And so this notion of clinical equipoise, meaning I don't know whether the drug will work or not, therefore I'm ethical in having a control group versus a treatment group, that's actually critical to designing the trial. You have to have some mechanism for being able to justify these kinds of trials. So with that said, blinding is another aspect of preventing yourself from somehow biasing the outcomes. In some cases, these are double-blind studies where obviously the patient doesn't know whether he or she is in the control group or the treatment group, but in addition, the investigators don't know either. And again, that's important because investigators can sometimes influence the study indirectly if they're not careful. And so it's very important to blind the data. In certain cases, it's not possible to blind the investigators. For example, if a surgical procedure is required and the investigators are doing the surgery, they can't really unlearn the surgery that they've done to a particular patient. So in certain cases, you're just left with a single blinded study. But double blind is typically the preferred. And again, the bias can be quite subtle. The Lincoln to Kennedy effect is something that we are all going to suffer from. Finally, once you engage in these clinical trials and you're done and you're ready to report, there is a standard for how to report the results. And the idea is to try to keep the standard relatively uniform so that you're comparing apples to apples across many different studies. And in the paper that you have assigned, this is the traditional way that medical journals like JAMA and New England Journal require you to report clinical trial outcomes.